Hello. Today we're going to be discussing epithelial tissues and talking about their structures, their functions, and the locations you can find them in the human body. Tissues are cells that perform specialized functions, and they're all grouped together by having similar structures and similar functions. When scientists study tissues, they refer to this as the field of histology. There are four basic tissue types: epithelial, connective, muscle, and nervous. Today we'll be focusing on the functions, the locations, and the distinguishing characteristics of epithelial tissues. If I had to describe the function of each of the primary tissue types with one word, I would say that epithelial tissues cover, connective tissues support, muscle tissues move, and nervous tissues control. Let's get into the characteristics of epithelial tissues. There's five basic characteristics that are only going to apply to epithelial tissues, not the other three tissue types. The first characteristic is that epithelial tissues have polarity. This means that the top of the tissue, or the more superficial part, has a different structure and a different function than the bottom part of the tissue. This top part is called the apical surface, and the apical surface is either exposed to the outside world, like with the epithelium on our skin, or it's exposed to the interior of a body cavity, so like the lumen or the inside part of a blood vessel. Conversely, the bottom part of the epithelial tissue is called the basal surface, and the basal surface is attached to connective tissues that support the epithelial tissue. The second characteristic of epithelial tissues is that they all have specialized contacts. These include things like tight junctions, desmosomes, and gap junctions. Here we have a cartoon image demonstrating those three types of specialized contacts. The first is a tight junction. Tight junctions are used to fuse cell membranes tightly together. You find tight junctions in places like the intestines. Desmosomes are similar in that they're also designed to fuse cell membranes together, but these are more like spot welds located in very particular areas between two cell membranes. You can find desmosomes in places like the skin. Lastly, we have gap junctions. Gap junctions are small tubular channels that are made of multiple proteins that all work together to create a small tube. And that tube allows small substances like ions and glucose to pass in between cells. Our third characteristic of epithelial tissue is that they are always, always, always supported by a layer of connective tissue. That connective tissue is called the basement membrane. And the basement membrane is made out of two very thin layers referred to as the basal lamina. This layer is closest to the epithelia, and the reticular lamina. This layer of the basement membrane is closer to the tissue that's underneath it. A key feature of the basement membrane is that it's completely non-cellular. This means that there's no cells that make up this tissue. The only thing that's in it is proteins that have been secreted by cells and some specialized carbohydrates like proteoglycans, water, and some other substances. But there's actually not any cells in the basement membrane. Whenever you see a word like this, proteoglycan, I want you to think of it as having two parts. The first part acts like an adjective, and it's describing the second part, which acts like a noun. So when we see the root glyc or gluc, I want you to think of sugars or carbohydrates. When you see this root prot or proteo, I want you to think of proteins. So since proteins is the adjective and glycans are the noun, this means that proteoglycans are carbohydrates that are modified with proteins. So they're carbohydrates that have little proteins stuck to them, kind of sticking out. The fourth characteristic really is two separate characteristics, but this is the fact that epithelial tissues are avascular but innervated. Whenever you see the prefix a in front of a word, I want you to think of that as meaning not. So this term avascular essentially means not vascular. And when we refer to something as being vascular or vascularized in the human body, what we mean is that it has a blood supply. So epithelial tissues are avascular, meaning they do not have a blood supply, but they are innervated, meaning they do have nerve endings. You can see that in this diagram of the human skin shown here. Skin is made of two layers. You've got the thin outer layer, which is called the epidermis, and this is made of our epithelial cells. And you've got a thicker lower layer that's deep to the epidermis called the dermis. The dermis is made of connective tissues, which we'll talk about another day. What you can see is that the blood vessels are exclusively in the dermis. They do not go up into the epidermal layer. However, you find nerve endings in both the dermis and in the epidermis. So when I'm trying to explain this concept that the epidermis is avascular but innervated, I'll generally ask students to think about a time when they've had a paper cut. You may have had a paper cut that was pretty superficial, meaning that it didn't go deep into your tissue. Perhaps it didn't even go deep enough for it to bleed, but it still really, really hurt. So paper cuts can be superficial enough that they don't bleed, but they're still painful because you still have free nerve endings in the epidermis. Our last characteristic is that epithelial tissues are really good at regenerating. So if you cut yourself or you have an abrasion or you have a burn, that epithelial tissue will grow back very quickly. This is dependent on adequate nutrients and good blood supply. But this can take place because there are stem-like epithelial cells in the very bottom sublayer of the epidermis that can go through cell division. A couple other ways of saying cell division is to say mitosis or cellular proliferation. Next, we're going to get into how epithelial tissues are named. This naming scheme is only going to be used for epithelial tissues. So when we get to connective tissues and the other ones, they're going to have their own naming scheme, and this will no longer apply. When we're naming epithelia, we have to use two different descriptors in the name. The first descriptor is based on the number of cell layers in the tissue. There's only two options here. The tissue is either made of a single layer of epithelial cells, or it's made of these cells that are more than one cell thick. So if the tissue you're looking at only has one layer of epithelial cells, we're going to describe that as simple. However, if the tissue has two or more layers, then we're going to describe that as stratified. The second descriptor that we have to use when describing epithelial tissues is what is the shape of the cell? There are three basic shapes that we're going to find. The first one is squamous. Squamous cells are flattened and they're very irregularly shaped. The nucleus is always also flattened. Cuboidal cells aren't perfect cube-shaped cells. However, they are approximately as wide as they are tall. Cuboidal cells are really easy to spot because they have very round, perfectly round nuclei. Lastly, we've got columnar cells and another category that's similar called pseudostratified columnar. And these shells are approximately column-shaped, meaning they're much taller than they are wide. And columnar and pseudostratified columnar cells have elongated nuclei. So notice what's happening here. The nucleus is always going to adopt the approximal shape of the cell. So if the cell is flat, the nucleus is flat. If the cell is as wide as it is tall, the nucleus is going to be as wide as it is tall. If our cell's stretched out, our nucleus is also going to be elongated and look like it's stretched out. Okay, let's get into some examples of epithelia. And we're going to start with the simple epithelia first. The functions of simple epithelia are really important, and you'll need to know them. They are absorption, excretion, secretion, and filtration. Absorption means that you're bringing a substance into the body. So an example of that is oxygen being absorbed from the air into your bloodstream via the structures in your lung. Excretion means you're getting rid of or you're eliminating something. So an example of that is that when you sweat, you excrete small amounts of substances called urea and lactic acid, and these are wastes. So sweating is actually a way to eliminate wastes from the body. Thirdly, we have secretion. Secretion is also a way of releasing something, but it's a way of releasing something useful. So the difference between excretion and secretion is excretion is getting rid of something we don't want, some sort of cellular or metabolic waste. Urinating is another example of excretion. However, secretion is when we're releasing substance that is useful to the body. So sweat can be useful to the body because it allows us to cool ourselves off when the sweat evaporates. So it's kind of interesting that sweat can fill both of these functions. Another example of secretion is when we secrete sebum or a type of oil from our oil producing glands. Lastly, we have filtration. Filtration is a way of purifying substances from the body. So the kidneys filtering waste from your blood is an example of filtration. Here we're seeing an example of simple epithelium that takes the squamous form. Remember, these cells are very flat, they're very irregularly shaped, and they also have flattened nucleus or nuclei. So when we look at kind of a top-down view of squamous epithelium, they look like this. But when we look at kind of a side-on view of histology tissue that is made from simple squamous epithelium, what we can see are our flattened cells here. And then we can see that they are supported by a layer of connective tissue. Some examples of simple squamous epithelium include the endothelium. Whenever you see this part, endo, I want you to think inside. So this is a thin layer of simple squamous epithelium that you find inside vessels, like blood vessels, lymphatic vessels, and other places in the body. Mesothelium is a type of epithelium that lines serous membranes. And serous membranes are very thin membranes that surround our organs internally. Next, we're going to discuss simple cuboidal epithelium. It's still one layer thick, but now we've switched to talking about the cuboidal cells. So these cells are about as wide as they are tall. And simple cuboidal epithelium is typically used for absorption and secretion. Here we have simple columnar epithelium. These cells are more column-shaped and have very elongated nuclei. They're tightly packed together, and these types of cells specialize in absorption and secretion. Now we have a very special type of cell called pseudostratified columnar epithelium. Notice that I've put simple in brackets. This is because this is a type of simple epithelium, but that descriptor isn't really used when giving the name. Pseudostratified columnar epithelium is how we describe it. What pseudostratified means is that it appears as if this tissue is stratified. Remember, stratified means that there's two or more layers. And if you look at this tissue, it looks like you've got a layer of nuclei up here, and then a second layer of nuclei down here. So it kind of appears as if there might be two layers here. But if you know your word parts and you know that pseudo means false, this means that it's falsely stratified or that it's not really stratified. So pseudostratified columnar epithelium wants you to think that it's stratified, but it's faking it. It's really a type of simple epithelium. If we were to move from one cell to the next, going from left to right, we'd see that here's a cell, okay, and then we move over, and then here's our second cell, and then we move over, and here's our third cell, and we move over, and here's our fourth cell. So it's really only one cell at a time in that layer. It's just that the cells have very irregular heights and widths. And so when they get packed together, it gives that appearance of being stratified when it's really not. The distinguishing characteristic of pseudostratified columnar cells is that they have cilia on them. And cilia are these little specialized structures, little cellular extensions. And they specialize in helping you to remove debris and mucus from the respiratory tract. So you find these cells lining your trachea and parts of your respiratory tree, like your bronchi and some of your smaller branches of the bronchial tree. And what they do is that they beat together in a coordinated fashion and help to move debris and mucus out of the lungs. So they help you to cough stuff up and get it out of your respiratory tract. If you smoke cigarettes or you know somebody that smokes, you might recognize that they have a really harsh, hacking, guttural cough. And this is due to the fact that smoking paralyzes the cilia. So smokers don't have the help of these little cellular structures synchronizing and sweeping the debris out of their lungs. So they have to cough much, much, much harder to try to move that mucus and move that debris out. Okay, that's it for simple epithelia. Now we're going to switch gears and we're going to talk about stratified. So remember, this means that there are two or more layers in the tissue. So these stratified epithelial tissues, like I said, have two or more layers and they regenerate from the basal side of the tissue. So remember, one of our characteristics of epithelia is that there is an apical side and a basal side. So the basal or the bottom part of the epithelia is where you find the cells that are proliferating. And then as these cells mature, they kind of move to the outer layers of the tissue. Stratified tissues are much more durable. So they're used for functions like protection, barriers, and sometimes secretion. Our first example is stratified squamous epithelium. Here you see a lot of different layers of epithelial cells. And one of the distinguishing characteristics of this type of tissue is that the outer cells on the apical surface have more of that typical flattened, irregular shape. Whereas the cells that are deeper and are along the basal surface tend to have more of a cuboidal or a columnar shape. And down here at the very, very bottom part of this tissue is where you find the epithelial cells that have that stem-like characteristic. That means that they act like stem cells, and stem cells are cells that are self-renewing. So they can divide and make more stem cells, but they can also make a daughter cell that will kind of mature into an epithelial cell, and then as it moves into these outer layers, it starts to take on some different shapes and different properties. And we'll talk more about that when we get to the integument and talk about the epidermis of the skin. Our second example of stratified tissues is stratified cuboidal. And stratified cuboidal cells are typically only 2 layers thick. I know they're showing 3 here in this diagram, but usually they're more likely to be 2 layers thick. These tissues, these stratified cuboidal tissues, are pretty rare in the body. You don't find a lot of examples of them. But some of the places that you will find them is in your sweat glands and in mammary glands. And these are the glands that produce milk. Fun fact, mammary glands are actually a modified sweat gland. So breast milk is actually a modified form of sweat. Kind of strange, isn't it? Uh, Here we have stratified columnar epithelium. These are also pretty unusual to find in the human body. You don't have that many examples of them. But you can find them in the pharynx, which is part of your throat, in the male urethra, and lining some glandular ducts. Here we have transitional epithelium. It's a type of stratified tissue, but we don't really use the word stratified when describing it. Transitional epithelium is called this because this is epithelium that you find in tissues that are very stretchy. And so the cells take on a different appearance depending on if the tissue is not stretched out or if the tissue is stretched out. If the tissue is not stretched out, the cells along the basal layer have more of a cuboidal or a columnar shape. And the cells on the apical layer, even though they look very cuboidal here, they can really vary in appearance. However, when these cells are stretched out, they take on more of a squamous, flattened out shape. And that's simply because as the tissue is getting stretched out, the cells are also getting stretched out. You find this tissue lining hollow organs like the bladder, for example. So when the bladder is really full of urine and it's stretched out and distended, that makes these cells transition into having more of a squamous shape instead of having more of a cuboidal or columnar shape.